At CRISPR Therapeutics, we are pioneering the use of CRISPR-Cas9 gene editing as a potential therapy for the inherited blood disorders, sickle cell disease and beta thalassemia. These diseases are caused by mutations in the beta subunit of hemoglobin, the molecule that carries oxygen in red blood cells. People produce different types of hemoglobin over their lifetimes. Fetal hemoglobin is produced leading up to birth and is gradually replaced with adult hemoglobin in the first few months of life. Only adult hemoglobin contains the defective beta subunit. As a result, newborns with these diseases do not experience symptoms until after hemoglobin switching has occurred. In sickle cell disease, the defective adult hemoglobin causes red blood cells to assume a deformed sickled shape. These sickled cells have difficulty circulating in narrow capillaries, giving rise to devastating pain crises. In beta thalassemia, on the other hand, symptoms arise because red blood cells have too little hemoglobin. These cells have poor function and only last a few days. However, due to natural genetic variations that scientists have identified, some patients continue to express fetal hemoglobin into adulthood and have less severe or no symptoms because fetal hemoglobin can substitute for the diseased adult hemoglobin. At CRISPR Therapeutics, our investigational CRISPR-Cas9-based therapy aims to mimic these natural genetic variations to raise fetal hemoglobin levels in patients. The therapy consists of isolating a patient's own blood-forming stem cells, editing the cells with CRISPR-Cas9 to increase fetal hemoglobin expression, and then returning the edited cells to the patient. The therapy begins with the mobilization and collection of a patient's blood stem cells. These cells are transported to a centralized manufacturing facility. There, through a process called electroporation, which temporarily opens cell membranes, the cells take up Cas9 guide RNA complexes that target a specific sequence in the DNA. The CRISPR-Cas9 gene editing process can then occur, resulting in a specific genetic disruption designed to increase fetal hemoglobin production. When the manufacturing process is complete, the patient's own blood stem cells, which have now been modified to produce fetal hemoglobin, are transported back to the patient's medical facility. Before receiving these edited cells, patients will receive chemotherapy to make space for the cells in the bone marrow. Patients will then receive the infusion of their own edited blood stem cells to repopulate the bone marrow, and following infusion, they will be closely monitored. Over time, the edited blood stem cells are intended to give rise to red blood cells that have increased levels of fetal hemoglobin with the hope of potentially reducing or eliminating patient symptoms.